So we'll be discussing infectious agents of diarrhea. And the first lecture, these are the learning objectives. So by the time I'm done with my discussion with you, I expect you to be able to tell me areas in the world where you have a higher prevalence of these agents of infection. So the epidemiology. It says you're signed out. Is it still recording? I also expect you to know the mechanism of disease, because as you'll find out, these agents use different strategies to cause infection. Having an idea of what is going on could explain how the patient presents clinically, and maybe what you should expect and what you should do. Then, how do you make a diagnosis? When and why do you send samples to the lab? What are the criteria for rejection? Is it every time that we receive a sample with from the patient who has diarrhea that the lab has to process the sample? So these are the points we're going to discuss. So this is my outline. So as a form of introduction, you should know that GI infections have a range of clinical manifestations. So the way the patient presents varies, okay? At times, the symptoms are associated with infection that is limited to the mucosal surface. So if you still remember from your physiology, the functions of the enterocytes, you can have an idea of how the patient will present. If the enterocytes functions are compromised, patient tends to have more of watery diarrhea. You don't have blood in the stools, and probably you won't have inflammatory cells because the infective process is limited to the surface, okay? Sometimes you can have direct invasion. So in this case, there's actual destruction of the enterocytes. So not only are the functions compromised, you may have some bleeding into the GI because now the integrity of the epithelial layer is compromised. So you may have a patient presenting with watery diarrhea initially, then it became bloody over time. So that can give you an idea of what is going on. At times, the microbes are not actually causing any problem, but the metabolites they produce, for example, toxins, these toxins may actually compromise the function of the enterocyte. In that case, we say that they are enterotoxin. At times, these toxins are actually lethal to the enterocyte. So we say they are cytotoxic. With enterotoxins, you may have just watery diarrhea, maybe some inflammatory cells, because some toxins may recruit inflammatory cells to the site of invasion. But when you have a cytotoxin produced, patients will eventually present with bloody diarrhea, as well as you find some degree of inflammatory cells when you examine the patient sample. Moreover, with some cytotoxins, with some enterotoxins, you may have absorption. So some patients may present with systemic symptoms as well as symptoms limited to the GI. So do you understand why we can have a spectrum of presentation? According to the WHO, diarrhea occurs when you have a patient passing liquid or watery stool at least 3 times in a 24-hour period. So that's the WHO working diagnosis. However, some medical scientists believe that you don't have to wait until you have 3 passage of watery stools 3 times in a day. At times, the patient will tell you that the nature of my stool has changed. So if the patient can tell you that something is wrong, you should believe the patient. Don't state that you're going to stick to rigid 3 times in 24 hours. Is that clear? At times, the diarrhea could be bloody or mucoid or bloody and mucoid. In that case, we refer to the stool as dysenteric stool. As you can see, you have a wide spectrum of agents that can cause diarrhea. Bacterial agents, parasitic agents. With parasitic agents, you can have the worms, you can have the protozoan. Then you can also have viral agents causing diarrhea. And each agent has a different mechanism. So clinically, the patient has diarrhea, but pathologically, the mechanism of the diarrhea varies from patient to patient and from agent to agent. In terms of epidemiology, diarrhea diseases are the second leading cause of death in children under 5 years. And it's also a leading cause of malnutrition in children under 5 years. The effect is more devastating in developing countries and in children in these countries below the age of 5. It is responsible for 1 death every 10 seconds in children under 5 in developing countries. Now, this data is not difficult to understand. Remember that children in these settings are already compromised nutritionally. Then you have a high prevalence of parasitic infections, like the parasitic worms, remember. And I told you that with the parasitic worms, they get to eat whatever the patient has first, before the patient gets to absorb anything. So these are patients that are already compromised nutritionally, okay? Then you now have a superadded infection that is making them lose nutrients. So that's why you have a devastating effect in these children in these regions. So this picture shows us the global mortality from diarrhea in children under the age of 5 in 2010. And as you can see, the hotspot countries are in the Asian and African continents. So you also have, apart from the hotspots, you can see that the death rate in African countries and in some Asian countries is also high. So how is infection acquired? Being a GI pathology, most agents are acquired through either contaminated food or water. There are some exceptions to the rule. For example, with Shigella species, you can have person-to-person contact, particularly when you use the bathroom and you don't wash your hands. It's possible to pass it on to someone else through a handshake, okay? You can also have transmission of some agent through fomites. You know what fomites are? So clinical presentation. Remember we said that the symptoms could be due to different underlying effects on the GI tissues. So when you have infections that are limited to the surface, like I said, you have a compromise in the function of the enterocyte. So you tend to have patients presenting with acute watery diarrhea, okay? If you have an invasive agent, patient tends to present with dysentery over time. And in some patients, with some parasitic agents, you can have chronic long-term infection, disrupting the function of the enterocytes. So the parasitic agent, Giardia species, can cause this. For acute watery diarrhea and invasive diarrhea, I've listed agents that are known to present clinically like this. So let's focus on some bacterial agents, starting with Vibrio cholerae. This is an important agent of diarrhea because you can have a pandemic infection. Vibrio cholerae are curved gram-negative rods. They are highly motile, okay? And they are fermentative, facultative anaerobes. They require salt for growth, so we refer to them as halophiles, that is, they love salt, high salt concentration. Now, they are divided into different serogroups and things like that. I am only providing this information for you to give you a background. I don't expect you to memorize this, okay? But this is important for epidemiology. If you have an outbreak, it's important to type the particular strain of Vibrio cholerae that is causing infection. So the serotypes that are very important to note are serotype 1, which is usually implicated in major pandemic. And it's associated with significant mortality in developing countries. Then you have serotype 013, which can cause a similar disease similar to serotype 01. So this can also result in a pandemic if care is not taken. This organism is found in estuaries and marine environments. So you don't have them in fresh water because we said they are halophiles. So it's where you have salt. So they are associated with salt water and associated with chitinous shellfish. The organism can have a free lifestyle, can multiply freely in water. And in 1 month, bacterial level can increase in contaminated water. Infection is acquired by consumption of freshly contaminated water or sea life from such contaminated water when they are consumed raw. You need a high infectious dose because these are the vegetative organism is the agent of infection, and the gastric acid content will destroy most of the ingested organism. So for somebody to come down with infection, the person must have really consumed a high dose of the organism. Now, the pathogenesis or the disease mechanism is toxin mediated. And what do I want you to note? The cholera toxin has a dimeric structure, okay? So you have the A subunit and the B subunit. The A subunit mediates the disease process. The B subunit mediates binding, so B for binding, okay? The B subunit, when it binds, results in the internalization of the toxin. Within the phagosome, the toxin is transported into the endoplasmic reticulum. At this site, the A subunit detaches and finds its way back into the cytoplasm to mediate the production of cyclic AMP. Now, the presence of increased cyclic AMP within the enterocyte is to reverse the absorptive function of the enterocytes. Remember that the enterocytes will absorb nutrients. So when the internal or intracellular concentration of CAMP, cyclic AMP, is high, instead of absorbing, they start to push out ions. So when these ions are pushed out, water will follow. So the patient tends to present with watery diarrhea, massive watery diarrhea, because the enterocytes are no longer absorbing nutrients. So clinically, the patient presents with what we call the rice water stool. The stool looks like water in which you have rinsed rice. So that's why we call it rice water stool. And it's diagnostic of Vibrio cholerae infection. You don't need a lab to back up your assumption, okay? In the lab, if you decide to send sample, it's mainly for public health data, okay? So we want to know what serotype is involved. That's not going to affect the management. You are not going to wait for my results to start treating the patient. Do you understand? So when samples are sent to the lab, it's mainly from the public health perspective. We need data to find out which particular serogroup is responsible for this current outbreak. It's not required for patient management. Is that clear? So in the lab, We know they are gram-negative curved bacilli. They're also said to be comma-shaped when you look at them under the microscope. They are curved, and they are motile. They have a polar flagellum. They are oxidase positive. Which other organism do you know that is oxidase positive? And the one that causes SDI? Listeria, very good. And they are also able to withstand a wide temperature range. Now, in the laboratory, we can isolate the organism selectively because remember, this is stool sample and there's a normal microbiota in the GI. So if we want to select for Vibrio cholerae, we use thiosulfate citrate bile salt sucrose agar, the TCBS agar. You can use McConkey. McConkey is non-selective, okay? On McConkey, they will show up as non-lactose fermenters because they can't ferment lactose. Is that clear? With the TCBS, you can differentiate between Vibrio cholerae and Vibrio parahemolyticus. Vibrio parahemolyticus will show up as non-fermenting because they can't ferment sucrose, non-fermenting greenish colonies. Whereas Vibrio cholerae can ferment sucrose, so you have this yellowish colony on TCBS. The virulence genes associated with Vibrio cholerae makes them pathogenic. So if they have these genes, the CTX, which mediates the cholera toxin production in the organism, and the TCPA, which mediates the attachment proteins to the enterocytes. Okay, so these are virulence genes that are associated with pathogenic strains of Vibrio cholerae. We move on to Escherichia coli. Now, from the renal block lecture, remember I told you that E. coli is a funny organism. You have strains that are normal commensals. You have strains that can cause UTI. You have strains that can cause meningitis in neonates. You also have strains that are adapted to cause infection within the GI. So this organism, Escherichia coli, is a complex organism, okay? Now, we can classify the strains that are associated with human GI infection based either on the clinical syndrome or the serotypes that we identify in clinical samples. Now, there are recognized strains, okay? Strains based either on clinical syndrome or based on serotypes, and you'll find out in a moment what that means. So the different strains that have been associated with human infection include the EHEC, which stands for enterohemorrhagic E. coli. From the name, how do you think the patient will present? So there'll be some form of dysentery, okay? It's also referred to as the sugar toxin E. coli, sugar toxin producing E. coli. This is because the toxin that is produced by these strains have a similar pathologic mechanism to that of Shigella toxin. Okay, so they are referred to as sugar toxin producing E. coli or enterohemorrhagic E. coli. You also have enteroinvasive, that's what the I stands for, enteroinvasive E. coli. From the name, how do you think the patient will present? Pardon? Yeah, that could be bloody diarrhea. What else? Yes, you may also have some systemic components because you now have an invasive strain, okay? Then you have enteropathogenic E. coli. You cannot guess the mechanism from the name, but just hazard a guess the likely group of patients that are affected by this strain. Pardon? Adults that are immune compromised, I agree. The P, as a medical student, the P was my key letter. This is prevalent in pediatric patients. Tends to cause infection in pediatric patients. So P, pediatric, enteropathogenic, okay? Then we have enterotoxigenic. So from the name, How will the patient present?
Diarrhea.
Watery diarrhea, very good. Then we have enteroaggregative E. coli. Yeah, they're forming clumps. So how do you think the patient will present? Remember, it's in the GI, not in the blood vessel. Yes. But what will eventually happen? They are forming aggregates, right? So what will happen to the enterocytes? Do you think the enterocytes will function normally?
No.
So what do you think will happen? Maybe you have watery diarrhea. Maybe there'll be some flatulence because they're not doing their job. The other microbes might, start acting on non-absorbed food materials and things like that, okay? It's also possible because they are aggregating, disturbing the enterocytes. You may have death of the enterocytes. So with time, you may have bloody diarrhea, okay? Then we have adherent invasive E. coli. How do you think the patient will present? Just a guess. We are still going to discuss them, but just hazard a guess from the name. Pardon? It's going to adhere to the surface of the mucosa, yeah? Maybe recurrent infections. Okay, we'll see. Anyway, so those are the list. For each strain, the pathologic mechanism is different. Is that clear? So let's see. Now, those strains of E. coli that cause infection are different from the normal microbiota. They have specific fimbrial antigens that enhance colonization of and adherence to the mucosa of the small intestine. Different strains exhibit different pathogenic mechanisms, like I've said earlier. So let's look at each strain, starting with enterotoxigenic. Now, enterotoxigenic is another strain that has a clue in the name. Let me put it that way. This is the usual agent of what we call traveler's diarrhea. So the T for traveler, the T for enterotoxigenic, okay? So this tends to cause traveler's diarrhea. So when you have people from non-endemic areas traveling to endemic areas, the map I showed you, those areas where you have a lot of deaths on the children under 5 years are endemic areas for these agents, okay? So for someone traveling from a non-endemic area to these endemic regions and coming down with diarrhea, the patient is telling you, oh, I went to India for a conference and I just came back. Now I have diarrhea. Think of enterotoxigenic E. coli first, okay? So the main pathogenic mechanism from the name is the production of an enterotoxin. Remember, I told you an enterotoxin is different from a cytotoxin. Enterotoxin will impede the function of the enterocyte in one way or the other. Whereas a cytotoxin is like a cellular bomb. It destroys the cell, okay? So the cytotoxin not only compromises functions, it destroys the cell completely. Now, these toxins are plasmid-borne. What does that mean? What? Yes, it's easy for them to transfer it to other non-toxigenic strains. Now, there are two types of toxins, the heat-labile and the heat-stable. Now, some strains produce just the heat-labile toxin, while others produce the heat-stable toxin, while some strains can produce both. So I guess it depends on the plasmids that are passed on to the different strains. The heat-labile toxin is similar in structure and action to the cholera toxin. So remember what the cholera toxin does. We have the A and the B unit. Remember, the B unit is for binding. The A unit has the catalytic function that increases intracellular count, okay? So the Cellular target of the heat label is similar to that of what we have with Vibrio cholerae. So it's the adenylate cyclase enzyme that is affected, and eventually you have increase in intracellular CAMP count. And remember, the effect of this is that the enterocytes no longer absorb, rather they are pushing out ions, and water will follow. With the heat-stable toxin, it has a different target, and the resultant effect is to have an increase in the cyclic GMP within the cell. The resultant effect is also similar to having high CAMP content within the cell. So the enterocyte no longer absorbs nutrients and ions, rather it keeps on pushing out ions, and water will follow. So how do you think a patient with toxigenic E. coli is going to present? Just watery diarrhea. Yeah, similar to cholera. We move on to enteropathogenic E. coli. Remember, I said this is prevalent in pediatric patients. Now, the pathogenic mechanism associated with the particular strain, as you can see, the organism is non-invasive. However, it closely attaches to the epithelial cells, okay? And in so doing, the organism affects the structure of these enterocytes. So instead of having this microvilli structure that increases the surface area for absorption, you have a stunting of these epithelial cells. They become like a pedestal upon which the organism sits. And of course, when you have a structure like this, the function of the enterocytes is no longer there. So they tend to present with watery diarrhea. So here you have an electron micrograph showing the pathology associated with enteropathogenic E. coli from a child who had a biopsy done. Now, let me quickly add that going for a biopsy is not your first line of management, because the biopsy is an invasive procedure, okay? Enterohemorrhagic or sugar toxin producing E. coli causes infection in infants and children. The impact and importance of this infection in public health is immense. And this is because this particular strain of E. coli is associated with what we call hemolytic uremic syndrome, in which the patient will present with acute renal failure. There is evidence of reduced thrombocytes in the blood, and then you have hemolytic anemia as well as other components to this presentation. Now, there are different strains of enterohemorrhagic E. coli, but the prototype, that is the one that you can use to explain what is going on with these strains of enterohemorrhagic E. coli, is E. coli 015H7. What does the O stand for? Somatic antigen, okay? And the H is flagellar antigen, very good. So we have different strains with different somatic antigen numbers, different flagellar antigen numbers, okay? But whatever the strain that you're dealing with, if it is enterohemorrhagic, E. coli 0157H7 is the poster boy for them. Is that clear? So that's why I'm not going to give you different names. If you understand what E. coli 0157H7 does, you can guess what the others are doing as well, okay? So this particular strain was the first to be linked to hemorrhagic colitis and hemolytic uremic syndrome in the early 1980s. And like I said, it's the poster boy for pathogens in this group. The common feature is the ability to produce sugar toxin. The sugar toxin is mediated by a bacteriophage. So what is the mode of transmission? Conjugation will be for plasmids. You were right the first time. Why were you doubting yourself? Okay. So there are two types of sugar toxin. You have the sugar toxin 1 and 2. The second one is more important, okay? And the main mode of action is to inhibit protein synthesis, okay? It does this by removing the adenine residue. The next picture shows this. It removes the adenine residue from the 60X ribosomal subunit in the bacterial agent. The resultant effect is that the bacteria can no longer synthesize proteins. Remember, you have, how does information move from DNA to RNA? What do you have? Transcription, right? Translation, right? And then protein synthesis, very good. And you need your ribosomes for protein synthesis. So if the function of these ribosomes are compromised, protein synthesis is arrested, okay? So that is the main pathogenic mechanism of this particular agent. Now, one thing I want you to note with enterohemorrhagic strains is that they bind to a particular receptor present in the enterocyte, the GB3. Now they are able to cause acute renal failure because this particular receptor is also present in renal tissue. So they can bind, the toxin can bind and destroy the renal cells, okay? It's cytotoxic in this case, and that's why the patient ends up with acute renal failure. Now, hemolytic uremic syndrome is prevalent in children age 10 and below. And in this group of children, if you suspect that the patient has enterohemorrhagic E. coli infection, do not rush to treat with antibiotics because studies show that it tends to worsen the clinical outcome in these patients. So don't rush to treat. Okay, as stated earlier, the syndrome, the hemolytic uremic syndrome is composed of at least these 3 clinical features. So you have thrombocytopenia, there's acute renal failure, and there is evidence of hemolytic anemia. Enteroaggregative E. coli can present with acute self-limiting diarrhea. Some patients may develop diarrhea for a long time, so it can also cause chronic diarrhea. The pathogenic mechanism is not well understood. Patients tend to present with watery diarrhea, with mucus, with or without blood. There could be abdominal pain, vomiting, and you can have low-grade fever. Now, this is the suggested or proposed mechanism of disease. So you have the planktonic bacteria. Planktonic means they are free living, they are floating all over the place. They are not attached to any cell, okay? These organisms will now form a clump and then attach to the mucosal surface. They start to produce biofilms, okay, and release toxins. The toxins may eventually destroy the enterocyte. That's why you may have bloody diarrhea later on in the disease progression, okay? And then you have biofilm formation, which also can induce damage to the epithelium and increase secretion. We have enteroinvasive E. coli. And because it's invasive, we know that the patient will present with bloody diarrhea. So it's a causative agent of dysentery in humans. The diarrhea due to enteroinvasive E. coli and Shigella is caused by the invasion and penetration of bacteria into enterocytes, leading to their destruction. Despite the similarities in invasion mechanisms and symptoms of the disease, the infectious dose of enteroinvasive E. coli is much higher than that of Shigella. Shigella is a highly contagious and infectious agent. The pathogenic features of enteroinvasive E. coli are virtually identical to that of Shigella. And the current model of pathogenesis involves these steps. You have epithelial cell penetration, lysis of the endocytic vacuole, intracellular multiplication, directional movement through the cytoplasm, and extension into adjacent epithelial cells. And this is what is explained here in this diagram. In terms of invasiveness, the organism may invade through the M cells, okay? Or you can have white blood cells finding their way between the epithelial cells. What happens is that the macrophages will ingest this organism. However, the organism evades phagocytic lysosome, what do you call it? You know, there's fusion of the lysosome with the phagosome. Very good. So the organism is able to prevent this from happening. Rather than being destroyed, it's going to replicate and escape from the macrophage and induce the destruction of the macrophage. Within the enterocyte, it's going to take over the protein machinery, protein synthesis machinery of the enterocytes. So the enterocytes, you know, Shigella are not motile, but when they invade the enterocyte, they commandeer the enterocyte to start producing what we call actin tails. These actin tails allow the organism to move from an infected cell to the adjacent non-infected cell. So once an enterocyte is invaded, it can be a self-perpetrating infection. You don't have to have fresh organisms coming from the mucosal surface before you can have invasion of non-invaded cells. Is that clear? Then we have the adherent invasive E. coli. This particular strain is linked to Crohn's disease. The pathogenesis of this group is not fully understood. So we have a lot of hypotheses, but we don't really know. The pathotype that is defined by the ability to adhere to and invade epithelial cells, replicate within the epithelial cells and macrophages. Studies on this particular strain shows that it can adhere to intestinal mucosa by binding to this receptor, carcinoembryonic antigen-related cell adhesion molecule 6, okay? So here you have a cartoon explaining how AIEC in Crohn's disease can, you know, lead to infection. So we've gone through the different strains, okay? So now I want to talk about your approach to diagnosis because this is very important. Now, from the description of the pathogenic mechanisms or the different strains that we have discussed, do you think a single laboratory test can detect or diagnose infection? No. Very good. So different pathogenic mechanisms, different approaches to diagnosis. So in terms of the sample you send to the lab, fresh stool sample is preferred, okay? The acute phase before you start intervening if you're going to use antibiotics. At times, this may not be possible. So a rectal swab can be sent to the lab or a surgically resected portion of the GI. In terms of how to get the sample to the lab, you may need a transport medium. Remember what transport media do? What do? They keep the organism viable until it is processed in the laboratory. So you can use buffered glycerol saline medium, or you can use Caribria. This is the most common. Now, in terms of trying to identify phenotypically and genetically, we can carry out stool examination. For example, is it watery stool? Is it dysentery? That gives an idea of the likely agents that could be involved in infection. Then the use of selective medium. So if we are using TCBS, we're probably thinking of which organism? What? Vibrio cholerae. We can perform biochemical analysis. We can perform infection of cultured cells, okay, to look at adherence pattern or ability to invade. We can use fluorescein actin stains, you know, when we talk of the enteroinvasive E. coli, or we can perform cytotoxicity assays. Remember that some toxins are enterotoxins, some other are cytotoxic. We can perform molecular assays using different assay approach. We can type. Remember, I told you with Vibrio cholerae, diagnosis is clinical. However, you need to send sample so that we know if there's an outbreak, we know which particular serogroup that we are dealing with. Then detection and typing future prospects. Now, MALDI-TOF is now top on the list of diagnostic tools that we have in the microbiology lab. It's a mass spectrometry assay. And the library against which you can compare your isolate with is expanding. So this is the top range tool that we have in the microbiology lab. Then you can use sequencing assays, or you can use single nucleotide polymorphism and MLST analysis of the whole genome. So this is telling you there's no one size fits all when it comes to making a diagnosis. However, from all the E. coli that we have discussed, remember I told you that the E. coli that is associated with hemolytic uremic syndrome, you know, you want to be sure what you're dealing with in this particular case because it can cause acute renal failure in children. So this particular strain of E. coli happens to have a peculiar metabolic characteristic. It cannot ferment sorbitol. So if you are thinking that this patient has infection due to E. coli 0157 H7, you can send samples to the lab. In the laboratory, you still remember the characteristics of E. coli. It's gram-negative rod. It's oxidase negative. It's a member of the Enterobacteriaceae. It's indole positive. You still remember. It's motile. And when you culture on McConkey, it tends to give you pinkish colonies because it can ferment lactose, right? Now, the other thing is after plating on McConkey, you also plate on what we call sorbitol McConkey agar. Sorbitol McConkey agar has essentially the same composition as the regular McConkey, but sorbitol has been added. Okay, so sorbitol can be fermented by non-pathogenic E. coli strains. So you have these pinkish colonies. These pinkish colonies, in this case, that is on sorbitol McConkey agar, these are the non-pathogenic strains. The pathogenic strains will show up as non-pinkish colonies. Is that clear? So we can use sorbitol McConkey agar to differentiate between the pathogenic and the non-pathogenic E. coli. Okay, so in terms of laboratory analysis, when you see a patient, there are certain questions you should ask yourself. Do I need to investigate this patient? So when you ask yourself that question, you're trying to find out if the identification is going to alter your management. Is the identification of the causative agent important from a public health perspective? So the patient has watery diarrhea, it's looking like rice water stool. Yes, you have to send sample to the lab because this has a public health significance, right? But the patient has just watery diarrhea, it's not looking like rice water stool. You're thinking of enteropathogenic E. coli. You don't really need to send sample to the lab, okay? Testing should, however, be considered under certain conditions. When the patient presents with diarrhea of more than a day's duration with bloody stools, because we know that strains that cause bloody stools are invasive or they produce cytotoxins. So in that situation, you want to be sure, what am I dealing with? If the patient has evidence of systemic involvement, so you have fever or the patient has symptoms of sepsis, if the patient is markedly dehydrated, if there is history of recent use of antibiotics, we are still going to discuss antibiotic-associated diarrhea. But for now, just know that if there is a history of use of antibiotics, you must send the stool sample to the lab. This is not when you start asking questions. Is that clear? If the patient has an underlying immunocompromised state, the patient just received renal transplant, okay? Or the patient is on cytotoxic therapy. So you have to send samples to the lab in this case. When identification of the causative agent is important from a public health perspective, so this cannot be overemphasized because we need the data. The data helps with surveillance, helps with intervention. So if it's going to be of public health significance, you don't have to question why you have to send the sample to the lab. Is that clear? Now, when you send samples to the lab, you don't throw stool samples at the lab. You don't say, do everything. Do everything does not make sense. So what are the things you should ask for? You've made up your mind that this sample must go to the lab. But what do you want the lab to do with the sample? So there are different tests that you can request for. If you have a food handler, somebody that is going to be dispensing food, you want to be sure that this person is not going to be dispensing infectious agents as well. So in this case, it's important to culture. Is this a carrier of Salmonella typhi? You want to be sure, okay? Giardia and Cryptosporidium, you have to screen for this. Presence of parasitic agents and cysts, you have to. So although the patient looks okay, the patient is coming for pre-employment review. In this case, you have to order for this test, but you don't have to do a toxin assay. In a child that is less than 5 years, you want to culture. In childcare attendants, you want to do everything just like with the food handlers, because these are people that are going to tend to babies and they're going to be feeding them. So you want to be sure that they are safe. Then regarding the other group of patients, and the tests that you can request for in the laboratory, I want you to note that when you have a patient who's had antibiotics, who's been treated with chemotherapeutic agents, who's had recent hospitalization, or the patient is elderly, you have to request for C. diff toxin assay. We'll discuss this later when we discuss antibiotic-associated diarrhea and why it's important to do the assay. So when we do a stool culture, why do we do it? We are looking out for pathogenic bacterial agents. And of course, since it says stool culture, stool will be the best sample. But under certain conditions, rectal swab, biopsy, that are transported in fecal transport medium can be submitted for culture. Duodenal aspirate, colonostomy samples, ileostomy samples, or sigmoid aspirate can all be sent. Now, who collects the sample? If your patient is mentally stable, can follow instructions, then the patient collects the sample. However, if you have a patient who has some mental compromise, okay, then the nursing staff or the physician can collect a swab and send to the laboratory. In situations where you need a duodenal aspirate, the physician will collect the sample and send to the lab. Now, the issue is the quantity of specimen. The textbook says at least 5 grams of feces. Nobody is going to be weighing feces, right? The patient certainly won't do that. So just tell the patient to have, a significant amount of samples sent to the lab. So when does the lab say no? If you have inappropriate or poor quality specimen for the particular assay you want the lab to perform. For example, you are querying entamoeba histolytica infection, acute. You're not querying whether the person is a carrier. You're saying this patient has amoebic enteritis, and then you are sending some stool to the lab. The lab is going to send it back, okay? It's not appropriate for the investigation you want to perform. If you have non-specific requests from the managing team, you know, I just said it, you don't throw stool samples at the lab. You don't ask us to go looking for something. Tell us what you think is wrong with the patient. Guide us so we know what to do. So the lab cannot run all the tests on the stool sample. So if your request is not specific, the lab is going to send the sample back. Well, if the sample has no label, we can't go looking for which patient has the stool sample. We can't. Samples collected more than 4 hours before submission to the lab, because it can affect the recovery or the isolation of certain pathogens from the stool sample. So there is a time interval. Now, if you have a sample container that is contaminated on the outside, we can't accept that in the lab. Or if it is leaking, it's going to be rejected. Note that stool samples should be examined and cultured as soon as possible after collection. So when it gets to the lab, we have to process within 30 minutes, particularly if you're suspecting amoebic colitis, or 4 hours after collection if culture is required. As stool specimens cool, there's a drop in the pH which will inhibit the growth of most Shigella and Salmonella species. So in conclusion, you can see that different disease mechanisms are deployed by different bacterial agents that cause diarrhea. In the lab, only adequately collected and transported stool specimens can be used for assays. And as a form of prevention, good personal, food, and environmental hygiene will prevent and control infection in the community. Any questions? Questions? So can we take attendance now? Sorry, it says I'm logged in. Sorry. Yeah.